Hello, welcome to Chapter 3, Sensation and Perception. In this chapter, we're basically going to look at all of our senses. The first part, your book will tend to explain the physical interactions, how the body perceives outside stimuli and translates it to nerve sensations, sends it to the brain. And then the second part of this chapter, we'll look at how we have perception and how we interpret the senses that we receive. First of all, let's take a look at sensation. This is the activation of receptors in the various sense organs. We have what's called sensory receptors, which are specialized forms of neurons. They're stimulated by different kinds of energy rather than by neurotransmitters. If you open up any of the PowerPoints, I embedded some videos. You can only see these if you can log into My Psych Lab. They're very short 1-minute videos, but unfortunately you can only access it if you have access to My Psych Lab. So you just punch in your login and password and you'll be able to see these. So you just click the link, and I don't know if it's going to work on this venue. I don't know if you can see that. And then there's just some students explaining what sensory abilities do you rely on.
Well, on a typical day, I feel that I rely on hindsight a lot because I tend to be commuting back and forth, I'm drive and just like I also feel that I'm in front of a book a lot and type. So eyesight is definitely something dependent for the day. But when it comes to specific settings, working as a babysitter, like the space and the smell is much more important than eyesight in this situation because, you know, if I taste the espresso side, it tastes burnt, or something smells a little off, goes a little foul, or something that needs to be assessed immediately because clearly quality isn't as best at that moment.
Okay, well, I'm not going to play the whole video, but what I want you to see is ask yourself, what sensory abilities do you rely on? We use different organs. We have our eyes, our ears, our nose, our skin, our taste buds, which all perceive the world around us. And the term transduction is turning outside stimuli into neural activity. And you can click and watch this video, short, like 1.5-minute video on the ABCs of sensation. And let me try to see if this works.
At the most basic level, you experience sensations. To make those sensations possible, energy must be converted into a signal that travels to your brain. This converting of energy is known as transduction. Light or electromagnetic energy is transduced so that we can see. Sound waves or mechanical energy are converted so that we can hear. Smell and taste result from the conversion of chemical energy. And then there's also the body senses, including skin sense, which is connected to touch, pressure, and temperature. Kinesthetic sense or body position is processed by the skin, joints, and muscles, and then the vestibular system contributes to balance and spatial orientation and originate and meaning.
Well, there you have it. When you talk about sensory thresholds, these are the differences between the two stimuli that's detectable 50% of the time. This was known as Weber's law, or probably Weber's law. Depends on what country, how you pronounce it. The absolute threshold is the smallest amount of energy. This was developed further by Gustav Fechner. The smallest amount of energy needed for a person to consciously detect a stimulus 50% of the time. There's a little link. You could actually do your own interactive experiment on Weber's law to see, how you can perceive differences in stimuli. For instance, here are some thresholds of sight. A candle flame at 30 miles on a clear, dark night, you should be able to see that. But if it's not clear or not that dark, you wouldn't be able to see. Hearing the tick of a watch 20 feet away in a very quiet room. I know there was some tours where people did a tour of the cave and they could hear their own heartbeat because it was so quiet. Smell, 1 drop of perfume diffused throughout a 3-room apartment. I have some students at school that use a lot of cologne. My nose really notices that. Taste, 1 teaspoon of sugar and 2 gallons of water. Touch, a bee's wing falling on a cheek from 1 centimeter above. So these would be examples of absolute thresholds. Now there's what's called subliminal stimuli. That's stimuli that are below the level of conscious awareness. You've heard of subliminal advertising. Basically, typically the influence of it influences automatic reactions, like it'll make you blink or something like that, rather than direct voluntary behavior. So it's probably not likely to make you buy a product suggested through advertising. With subliminal perception, it basically will create us, or will cause a process by which subliminal stimuli act upon the unconscious mind, influencing behavior. But like I said, it's unlikely to affect direct voluntary behavior. It's not going to make you buy something you don't want. Habituation and sensory adaptation. Habituation is the tendency of the brain to stop attending to a constant, unchanging information. For instance, have you ever walked into your house and you're like, oh, it stinks in here. And after a few minutes, you don't even notice it. But unfortunately, anyone who comes visits you will also notice that stink. So your senses adapt to real strong sensory information if it doesn't change over time. So this is a tendency of those receptor cells to become less responsive to a stimulus that's unchanging. For instance, if you ever had some real strong tasting food, that strong taste fades over time. So your brain is programmed to notice changes in stimuli, but after a while ignores unchanging information. Isn't that kind of cool? Oh, this word, microsaccades. It's a constant movement of the eyes. There's tiny little vibrations that people do not notice consciously. For instance, you notice how I told you your body tends to ignore things that are the same, unchanging information. That's different though for your visual, which is a good thing, because your eyes can still notice changes. It keeps the eyes from adapting to what they see. So there's like a constant movement of the eyes, and you're constantly getting a sensory visual stimuli, or sensing visual stimuli. With the eye, it's an amazing organ. Studies brightness. The 3 things that it's going to, when you perceive light, you're perceiving brightness, color, and saturation. Let's first look at brightness. Brightness is determined by the amplitude of the wave, or how high or how low the wave actually is. So low waves are dimmer, higher waves are brighter. Next is the color or the hue, which is determined by the length of the wave. If it's a long wavelength, they're found at the red end of the visible spectrum. And then shorter wavelengths are at the blue end. You know the color spectrum, the Roy G. Biv. Well, the R side is more longer wavelengths. The Biv are shorter wavelengths. Next term is saturation. Saturation is the purity of the color. When you mix in black or gray, it lessens the saturation. There's a cool little video here on visible spectrum. I'm not going to show it, but you can click it if you watch the PowerPoint version of this. Here's the visible spectrum. Notice Roy G. Biv plays only a small part of the visible spectrum. We also have gamma rays, X-rays, radio waves. You probably noticed how we all switched, that was a few years ago, how we all switched from regular TV waves, which use the FM stations, to digital. And those waves are more compressed, so you could have actually more stations over the air. That's why you have more TV stations over there now than what it used to be. However, those signals are not as strong, so you need a better antenna to pick those up. Structure of the eye. There's actually a little, these are all the parts of the eye. The eye is a very intricate, complex organ. If you want, you can study up on these, and on the next slide, You can test yourself on the structure of the eye terms. We have the cornea, which is a clear membrane on the outside of the eye that protects your eye, focuses on most of the light coming in. This is where you have your photoreactive keratectomy and laser-assisted in situ I haven't brushed up on these words. Keratomylusis, or LASIK vision, improving techniques that make small incisions in the cornea to change the focus in the eye. So if you ever have corrective surgery on your eyes, LASIK surgery, what they do is they mess with your cornea, so you don't need contacts or glasses. Sometimes that's just temporary. After a few years, you still need another surgery or something to maintain that vision. The aqueous humor, this is a visual layer below the cornea. It's clear watery fluid that's continually replenished. It supplies nourishment to the eye. Then there's a pupil, which is a hole which the light from the visual image enters the interior of the eye. You have the iris, the round muscle, the colored part of the eye in which the pupil is located. And this can be controlled. It can change the size of the pupil, letting more or less light into the eye. If you ever notice, if you shine a light in someone's eyes, the iris changes. You'll see less pupil. If the darker the room, the iris opens up and you'll see more pupil. The lens is another clear structure behind the iris. And then the body makes what's called a visual accommodation. This would be a change in the thickness of the lens as the eye focuses on the objects that are far away or close. Just like if you use a camera, you zoom in, zoom out, your eyes do the same thing. The vitreous humor is a jelly-like fluid that also nourishes the eye and gives it shape. Now, if you have nearsightedness, that's also called myopia. This is the shape of the eye, causes a focal point to fall short of the retina. If you have farsightedness, the focal point is behind the retina. This next image will help you see nearsightedness and farsighted. This is why I'm wearing my cheap readers. Let's see, nearsighted is, I always get these two mixed up, but I can see things far away, but I can't see things up close, so I have to have these readers. And that's because my eye does not focus well on things that are close. The retina is the final stoplight for the light in the eye. It contains 3 levels: so your ganglion cells, bipolar cells, and your photoreceptors. Cool little video here, about 2 minutes long, that helps you understand rods and cones and how your body can perceive light and transmit it to the brain. The rods are the visual sensory receptors found at the back of the retina. They're responsible for non-color sensitivity to low levels of light. The cones, on the other hand, they handle the visual sensory receptors found at the back of the retina. They're responsible for color vision and the sharpness of your vision. And we all have a blind spot. And this is the area in the retina where the axons of the 3 layers of retinal cells exit the eye to form the optic nerve. This is a part that's insensitive to light. Now the tricky part, your brain actually adapts to this blind spot. We tend to fill in what we're not seeing. But here's a little experiment on the parts of the retina. And here's where this here is where light passes through ganglion and bipolar cells until it reaches and stimulates the rods and cones. The nerve impulses from the rods and cones travel along a nerve pathway to the brain. On image B, on the right of the figure, is a photomicrograph of the long, thin rods and the shorter, thicker cones. The rods outnumber the cones by a ratio about 20 to 1. Now, image C is a blind spot demonstration. What you want to do is hold the book in front of you, close your right eye, and stare at the picture of the dog with your left eye. Slowly bring the book closer to your face, and the picture of the cat will disappear at some point because the light from the picture of the cat is falling on your blind spot. If you cannot seem to find your blind spot, try moving the book more slowly in and out until the cat disappears. Now, light falling on the left side of each eye's retina from the right visual field, shown in yellow, will stimulate a neural message. that will travel along the optic nerve to the thalamus, and then on the visual to the visual cortex in the occipital lobe of the left hemisphere. Notice that the message from the temporal half of the left retina goes to the left occipital lobe, while the message from the nasal half of the right retina crosses over to the left hemisphere. The optic chiasm is the point of crossover. The optic nerve tissue from both eyes joins together to form the left optic tract before going on to the thalamus and the left occipital lobe. For the left visual field, shown in blue, the messages from both right sides of the retina will travel along the right optic tract to the right visual cortex in the same manner. The interesting thing about this is your brain is actually picturing images perceived in our brain as upside down and reversed. But don't worry, our brains compensate for this. So we're always viewing the world upside down and reverse in our brain, but our brain automatically corrects that. Isn't that nice? Otherwise, we'd be seeing everything upside down. How the eye works. There's what's called dark adaptation. And this is the recovery of the eye's sensitivity to visual stimuli in darkness after exposure to bright lights. So for instance, if you're walking around in the dark and some kid shines a flashlight in your eyes, suddenly you have what's called night blindness because your eyes just adapted to that bright visual stimuli. On the other hand, if you walk around in the dark for a long time and you don't have any bright lights shining in your eyes, you have what's called light adaptation, and this is where your eye sensitivity to visual stimuli and light after exposure to darkness. I'm sorry, that's where the person shines a flashlight in your eye. But dark adaptation, you notice when you walk around in the dark, you can start seeing pretty well. You're actually focusing on a different part of your eye, and you're able to see well in the dark. Sometimes when I go camping, I shut off my flashlight and I let my eyes adjust to dark adaptation. And I can see better without the flashlight than with, if I just let my eyes adjust to the dark until some kid shines a flashlight in my eye. Trichromatic theory. This is a theory of color vision that proposes 3 types of cones. That we have red, blue, and green. This came from Thomas Young. Later on, Hermann von Helmholtz pushes this theory. The second theory is what's called the opponent process theory. This is theory of color vision that proposes 4 primary colors with cones arranged in pairs, red and green, blue and yellow. And an experiment that helps push this theory is what's called the after images theory. If you stare at an image long enough, and then you stare at a white wall or a white piece of paper, that image still is present. You might want to try that with the next slide. But images that occur when a visual sensation persists over a brief time, and even after the original time the stimulus is removed. So we'll try that in a second. This is called lateral geniculate nucleus, LGN, of the thalamus that creates this function. So take a look at this image. I want you to stare at the white dot in the center of this oddly colored flag for about 30 seconds. Stare at the white dot. Now look at a white piece of paper or a white wall. You'll see the image of the flag on the white wall. Pretty cool, huh? The expected colors of the American flag show up, so you're actually seeing it in red, white, and blue. They're also the primary colors that are the opposite colors in this picture. And they provide evidence for the opponent process theory of color vision. So these are the opposite colors of the flag. And your brain actually fills that in when you look at the white wall or white piece of paper. Pretty cool, huh? Next is color blindness. Whenever you have a driver's test, you probably notice some of these tests they'll give you. A monochrome color blindness is a condition in which a person's eyes either have no cones or have cones that are not working at all. Now this would be very important in being able to see when you're driving because you want to be able to see signs and be able to read the signs and a color contrast in the signs. Red-green color blindedness, either the red or the green cones are not working. Let's see what else I want to cover on this. That would be called dichromatic vision, where you have red-green color blindedness. So why are most people with color deficiency men? Usually it's related to what's called sex-linked inheritance. So it's attached to chromosomes that determine this, that also determine the sex of a person. The gene for color deficient vision is recessive. Therefore, for males, both parents would need this recessive trait to be passed on for females. However, for males, this only needs to be inherited. They only need to inherit one recessive gene to be passed on. And here's a kind of a cool little concept map on the sense of seeing. I'm going to open it up so you can see. But you can see like the progression of seeing. You have the stimuli of light, and there's a physical stimulus, and as a form of electromagnetic radiation processed by the eye. We have the cornea, pupil, lens, the retina, and then there's our photoreceptors and rods and cones, and that translates it to our brain. The other part of seeing is the retinal receptor, rods and cones. You have our periphery of peripheral vision, and we also have our colors that we are seeing as well. Kind of cool little concept map. Okay, this, when you take a driver's test, I printed my slides out in black and white, and I was totally unable to see these numbers. You need to have this in color to see this, but you should be able to see the number 8 on the left and number 96 on the right. Then that shows that you have a good sense of color. You're not colorblind. Okay, next, wavelengths in the ear. This has to do with basically our body's ability to interpret sound or vibrations of molecules in the air around us. How do we do that? We do that by sensing these things: wavelength, amplitude, purity. Wavelength can be interpreted as a frequency or pitch, high, medium, or low. And amplitude is interpreted as volume. So when someone turns up the volume, they're turning up the amplitude of the waves. And then purity of the waves is interpreted as timbre, which is the richness or tone of the sound. And hertz is just a measurement of cycles or waves per second. This would be measuring your frequency of your wavelength. This basically shows amplitude. This would be a louder sound. And notice a shorter wavelength would be a softer sound, higher pitch. Or a lower wave is softer. So look at your two sound waves. The higher the wave, the louder the sound. The lower the wave, the softer the sound. If the waves are close together in time, which is high frequency, the pitch will be perceived as a high pitch. Waves that are farther apart, which are low frequency, will be perceived as having a lower pitch. This shows decibels of various stimuli. A decibel is a unit of measure for loudness. Psychologists study the effects that noise has on stress, learning, performance, aggression, and psychological and physical well-being. So if you're listening to music with your headphones turned to the highest volume, that's causing a pain threshold, but also it's causing more stress, probably less likely to study. But if you have a quieter environment, you're probably more psychologically able to learn. I notice soft music without words helps me to study. But if I have music that I really like, where I sing along, then all of a sudden my brain is distracted and not able to focus on the stimuli up ahead. Next is the structure of the ear. We have the external ear, or the outer ear, which is the auditory canal, which is a short tunnel that runs from the pinna, which is the outside part of your ear, to the eardrum, which is your tympanic membrane. Next is the middle ear. The middle ear is the eardrum, which is the thin section of skin that tightly covers the opening into the middle part of the ear. When sound waves hit the eardrum, it vibrates and causes three tiny bones in the middle ear to vibrate. These three tiny bones are named after their shape: hammer, anvil, and stirrup. So if you look, if it's shaped like a hammer, it's the hammer. Okay. Next is which is called the inner ear, and this is where it takes it perceives the outside vibrations and it actually converts it into signals to your brain. The cochlea is a snail-shaped structure of the inner ear that is filled with fluid. The organ of Corti rests in the basal membrane, contains receptor cells for the sense of hearing. The auditory nerve then is a bundle of axons from the hair cells in the inner ear. So in other words, there's these little tiny cilia that somehow translate the signal into neural messages. And if you do this, you could do check your understanding of the parts of the ear. It's a cute little thing you can test yourself on different parts of the ear. So you can check that out at another time. This is the structure of the ear, and that we just went over. So we have some nice visual images. Next, perceiving pitch. Pitch is a psychological experience of sound that corresponds to the frequency of the sound waves. The higher the frequency, higher frequencies are perceived as higher pitches. Place theory is a theory of pitch that states that different pitches are experienced by the stimulation of hair cells in different locations on the organ of Corti. Frequency theory is a theory of pitch that states the pitch is related to the speed of vibrations in basal memory. Okay, a couple things I want to talk about. With frequency theory, this came up from Ernest Rutherford back in 1886. And I just talked about two different theories. There's what's called the place theory, okay, and then frequency theory. And the question is, which of the theories is right? Both frequencies actually work. Both theories actually work. Frequency theory works well for low pitches. Place theory works well for moderate and high pitches. Now there's another theory called the volley principle. This theory of pitch states that frequencies from 40 hertz up to 4,000 hertz cause the hair cells or auditory neurons to fire in a volley pattern, or they take turns firing. This is the third theory by Ernest Weaver and Charles Bray. And actually this one works too as well, but just under these hertzes. Okay. Now, you've had people have hearing problems. A hearing impairment is when you have difficulties in hearing. There's partially impaired, and where people might have like a hearing aid in their ear. And then there's what's called totally impaired, where they can't hear at all. This may be caused by problems with the mechanics of the outer or middle ear, like a damaged eardrum, which can prevent sound waves from being carried to the middle ear. Or you could have damages to the bones of the middle ear, where sounds cannot be conducted from the eardrum to the cochlea. Then there's what's called a hearing impairment, where the problem lies in the inner ear, and the damage of the auditory pathways and cortical areas of the brain will impair a person's ability to interpret the stimuli of sound. And there's a little concept map on the hearing sense that you can use. Now, if the problem is the inner ear problem, you can have what's called a cochlear implant. A microphone is implanted just behind the ear that picks up sound, and there's a speech processor that selects and arranges the sound picked up by the microphone. And the implant is a transmitter and receiver converting signals to electrical impulses. This is kind of the miracle of today's technology, where people who were totally impaired are able to hear again. Then this is collected by an electrode array in the cochlea and then sent to the brain. So people who are totally impaired can have the ability to hear again. So this is an example of an inner ear processor. Let's talk about taste buds. Taste buds actually use the combined organs of taste and smell. The taste buds involve receptor cells in your mouth, not only your tongue, but your upper mouth, your walls of your cheek, all contain taste buds. The term gustation is the sensation of taste. This nerves in the tongue's deep tissue, and then figure B, taste buds location inside the papillae. And then C is the micro photograph of the surface of the tongue, yuck, showing the two different sizes of the papillae. Now those little lumps there, those aren't the taste buds. Actually, the taste buds are underneath those little lumps. And this is part of your brain. The gustatory cortex is found in the anterior insula and frontal operculum. The insula is an area of cortex covered by folds of overlying cortex, and each fold is an operculum. In the coronal section of the human brain above, the gustatory cortex is found in regions colored in a light red. If you notice, a huge amount of resources in your brain are put towards taste and smell, which must have been a pretty important part of our evolution and survival as the species. There are 5 basic tastes: sweet, sour, salty, bitter, brothy, or umami. There's a little video on taste you can view if you want. And what I mentioned earlier is Hans Henning in 1916 came up with these 5 basic tastes. But the tastes are one of our earliest developed senses that utilize both taste and smell. Have you ever had your nose stuffed up and tried eating food and you noticed food didn't taste as well, or you didn't taste as effectively? That's because the olfactory sense plays a big part in taste as well. So let's talk about the olfactory sense, our sense of smell. Basically, areas of the brain located above the sinus cavity and just below the frontal lobes that receive information from the olfactory receptor cells. There's at least 1,000 olfactory receptors in our brain. So a huge part of our brain is used towards sense of smell. And there's a nice little concept map on smell that you can view as well. This gives you an image. Smell comes in through our nose, and then we have the cilia of olfactory receptor cells, and the signal goes into the olfactory bulb and goes into the brain. The term somaesthetic senses is where the body senses the body senses consisting of the skin senses, the kinesthetic sense, and the vestibular senses. Okay, so soma is a term which means body, aesthetic means feeling. Two key things I want to talk about with this has to do with there's sensing pressure. Also, it has to do with sensing pain, and vestibular has to do with balance. Kinesthetic has to do with moving, being able to walk and move. So the skin senses touch, pressure, temperature, and pain. We have receptors in our skin that can pick up these senses. And then there's what's called the gate control theory, where pain signals must pass through a gate located in the spinal cord. For instance, if you ever had intense pain in your back and it feels very, very painful, your brain or your sense signals are being sent to your vertebrae and then up to your brain saying you're going through a lot of pain. If somebody massages that area, gate control theory is the physical stimuli is actually shutting off the pain signal to your brain. So that shows why a massage might help with pain. Also, you can use visual techniques or psychological techniques to help manage the pain as well. And there's a little video here on the gate control theory of pain. Next is a cross-section of the skin and its receptors. This basically helps you understand how there's pressure-sensitive receptors, and then there's pain-sensitive receptors that send signals to the brain. Okay, next is a kinesthetic sense. This is the sense of the location of the body parts in relation to the ground. So when you're walking, you're using your kinesthetic sense. Otherwise, you'd probably fall. Your proprioceptive receptors also play a part in this. And here's a little concept map to help you understand the somatic senses. You know, your vestibular senses has to do with movement, balance, and body position. The term sensory conflict theory is an explanation of motion sickness in which the information from the eyes conflicts with the information of the vestibular senses. I don't know if you've ever been on a boat, you might have what's called dizziness, nausea, or physical discomfort. That's basically your eyes are having a hard time figuring out the information based on what the vestibular senses are. For instance, if you're on a sailboat and you're rocking a lot in the waves and you're feeling woozy, it's very important to stay topside so you can keep your bearings with your eyes can keep bearings on the movement of the boat and the movement of the water, and you're less likely to get seasick. If you go down below in the boat, you're more likely to get seasick because you can't see the rocking motion, and you lose your frame of reference. Just a little advice if you ever go on a boat. Stay topside if you get sick. Okay, perception. This is your brain's way of interpreting the information that you're receiving from your sight and from your somatic senses. This is a method by which the sensations experienced at any given moment are interpreted and organized in some meaningful fashion. The term size constancy has to do with the tendency to interpret an object as always being the same size regardless of its distance. And I'll give an example in a minute here. The shape constancy is the tendency to interpret the shape of an object as being constant, even when its shape changes on the retina. And then there's brightness constancy. It's the tendency to perceive the apparent brightness of an object as the same, even when the light conditions change. Let me give you some examples. This is an example of shape constancy. Notice you can see like it looks like a door opening, and the second one, your brain is actually seeing the triangle and the circle as turning, or the triangle is turning. And then the bottom one, you see, it looks like a hat, but your brain is able to figure out those shapes and figures. Now, some Gestalt principles can be applied here. Figure ground principle is where the tendency to perceive objects or figures as existing on a background. This is where you look at three-dimensional figures and you can start understanding. Then there's what's called reversible figures, where visual illusions in which a figure and ground can be reversed. Well, hi Cooper, come over here, doggy. This is my dog, Cooper. Oh, I gotta change it. Hey, Cooper. This is my daughter, Sophie. She's kind of waking up too. Hi, Sophie. Okay, let's get back to class here. Figure ground. Okay. Let's see. Here's an image of what's called the Necker cube. This is what's called an ambiguous figure because it's not clear which shape can dominate. So this could be the front of the cube right here, or you could image it with this as the front of the cube. Your mind can play little tricks on you like that. This is another image of a figure ground illusion. You can see it as two people facing each other, or you could see it as a, I don't know, a bird bath or a vase. Next image. is what's called proximity. It's a tendency to perceive objects that are close to each other as part of the same grouping. And then there's similarity, which is a tendency to perceive things that look similar to each other as being part of the same group. Closure is a tendency to complete figures that are incomplete, and continuity is a tendency to perceive things as simply as possible with a continuous pattern rather than complex broken up pattern. This is where you might see your brain kind of fills in the blank. And so you might not even see a total image, but your brain kind of connects the dots and helps you see a pattern. It's kind of complex part of perception. Contiguity is a tendency to perceive two things that happen close together as being time related. Now with contiguity, the image is not shown because it involves nearness in both space and in time. The best example of contiguity is ventriloquism, where there's that illusion of the voices coming out of the puppet when actually it's coming out of the person. That would be an example of contiguity. So this shows proximity or nearness. Notice how you can see the difference. Similarity of the group, so your brain automatically groups these by color. Closure is how, although it's a dashed line, we tend to fill in the dashes and perceive a circle or a square. Continuity is where we take the dots and we actually see images from that pattern. And in common region, we see similarities and differences. Now next is depth perception. This is the ability to perceive the world in three dimensions. We have what's called monocular cues. These are cues that you can pick up even if you're using only one eye. Okay, we can see linear perspective, the tendency for pair lines to appear as converging on each other. And then we see relative size, and that's perception that occurs when objects that a small person expects to be of a certain size appear to be small and are therefore assumed to be much farther away. And we'll show some examples here. Overlap is the assumption that an object that appears to be blocking part of another object is in front of the second object and closer to the viewer. And then the aerial perspective is a haziness that surrounds objects that are farther away from the viewer, causing the distance to be perceived as greater. And then there's what's called texture gradient, which is a tendency for textured surfaces to appear to become smaller and finer as distance from the viewer increases. Next is motion parallax, which is the perception of motion of objects, in which close objects appear to move more quickly than objects that are farther away. And then there's what's called accommodation, the brain's use of information about the changing thickness of the lens of the eye in response to looking at objects that are close or far away. Here are some images. A is what's called linear perspective. B, so notice it looks like the two lines are converging as it goes far away. Next is texture gradient. Notice you can see the texture easier on the closer figures and the farther figures. Aerial or atmospheric perspective. Notice as it goes farther, it looks hazier. The items closer are a lot clearer. And then relative size. Notice the flowers up close. We can really focus on the details. In the flowers down farther are actually the same size, but in our perception are smaller, to give us an idea that it's farther away. Next are binocular cues. These are cues that we need both eyes to understand. Since our eyes are farther apart, we actually get two different images from each eye. Convergence is the rotation of the two eyes in their sockets to focus on a single subject, resulting in greater convergence for closer objects and lesser convergence if objects are distant. Binocular disparity is the differences in images between the two eyes, which is greater for objects that are close and smaller for distant objects. So if you take your finger and you follow it with your eyes, notice you have a little more, you have two fingers before you as you move it closer. Those are binocular clues because you have a disparity of images from both eyes. And it's just an image. It shows as the image gets closer, you have what's called binocular disparity. Next are perceptual illusions. The Hermann grid shows the effect that's possibly due to the response of the primary visual cortex. The Muller-Lyer illusion is an illusion of line length that's distorted by inner turning or outward turning corners of the ends of the lines, causing lines of equal length to appear different. You can try this experiment of the Muller-Lyer illusion by clicking this link here on the PowerPoint. This shows the Hermann grid. What you want to do is stare at the entire image as a whole, and you'll notice little black blobs or triangles in between. You'll be like gray blobs or diamonds, and they'll fade away when you look directly at the intersection. Kind of a freaky little illusion. Next is what's called the Muller-Lyer illusion. Believe it or not, in figure A, both of these lines are exactly the same length. But because of the images protruding from the outside, it gives us an illusion that one is longer than the other. Same thing with both of these. The line is the same length if you measure them, but the illusion is that this one's longer. Next is the moon illusion. The moon horizon appears to be larger when it's in reference to, let's say, the tree or anything like that. But when the moon is high in the sky, it looks smaller. That's another illusion. There's illusions of motion, which is called the autokinetic effect. It's where a small stationary light in a darkened room will appear to move or drift because there's no surrounding cues to indicate that the light is not moving. I might freak you out and say, why is that light moving? Next is the stroboscopic motion, where it's seen in motion pictures in which a rapid series of still pictures will appear to be in motion. Next illusion of motion is the phi phenomena, where lights turned in on a sequence appear to move. Where there's rotating snakes are due to, in part, of eye movements. We're going to show these images. The enigma is due to microsaccades, which means your brain is constantly re-scanning, and it looks like the parts of the picture are moving. So you can try this interactive experiment. This is kind of a cool one, where you can, it's on ambiguous figures, where they'll lead you with a few images, and then your tendency is to pick out a certain image on the final image. It's a pretty cool thing. You might want to check that out. This link right here. Okay, stare at this picture, and after a while it looks like there's rotating snakes. It looks like it's rotating. That's because your brain is constantly rescanning and rescanning, and it actually looks like it's moving. Pretty cool, huh? This is another one. It's actually a painting, 1981 painting from Sia Levant called Enigma. And if you look at it, it looks like it's rotating. Pretty cool, moving picture. That's not really moving. It's an illusion. This shows the Ames room illusion. This kid is much taller than his dad, but the room creates that illusion. Pretty cool stuff here. But your brain tries to make sense of the picture, and it's pretty cool. The perceptual set, this is where your brain creates perceptual expectancy, the tendency to perceive things in a certain way because previous experiences or expectations influence those perceptions. We have what's called top-down processing. This is the use of pre-existing knowledge to organize individual features into a unified whole. The best way to understand top-down processing is when you're doing a puzzle and you have the picture of on the box top, so you have a pre-existing image of what this puzzle is supposed to look like. Okay, and that's top-down processing. Now, bottom-up processing is if you're putting together a puzzle and you don't have the box top with the picture, and you're trying to piece it all together and figure out what it's supposed to be. Of course, that's a lot harder than top-down processing where you have pre-existing knowledge. Okay, take a look at this picture. What do you see? Okay, some of you might see a young lady looking away. Some of you see an old lady looking down. Take a look at this next slide. Do you see the old woman looking down? And on the right, you see the young woman looking away. This is how your brain interprets the first drawing and the second drawing differently. Like I said earlier, bottom-up processing is the analysis of smaller features to build up on the complete perception. Here's a concept map of perception to help you understand perception. Next, take a look at this image here, the Devil's Trident. You probably are able to see three different, what's that called? Three different prongs. However, this is actually an image that can't really be created in real life. If you follow the lines, it couldn't be really created into a real image. This is just optical illusion. Take a pencil, grasp it in the middle, shake it, wiggle it up and down like this. And actually it works better with a pencil. But if you do that, it looks like the pencil's rubber and it's bending like this. It's pretty cool. Magicians often use this type of imagery or illusion to blend solid objects. There's what's called end-stop neurons. In the visual cortex, it's sensitive to motion and edges. These neurons respond differently. If an object is bouncing, it causes a perception of blending. So our brain always makes adaptation. This is the end of chapter 3. I know this was a very long presentation, 52 minutes. Thank you for staying with me. There'll be a quiz on this, so go ahead and open up the quiz and have a great day.